Today's question is it's quite worrying because people are quite fearful still of the clonal evolutionary theory, which basically to put it briefly, the whole idea that somatic gene mutations cause cancer. And if I could start off by saying here, less than 1% of gastric cancers to start off with here, because the question was, is stomach cancer hereditary? Less than 1% of all gastric cancers are hereditary. Less than 1%. And less than 10% of all cancers in the world, are linked to your genes. And of the remaining 90%, the mutations are still late epiphenomena, meaning that these mutations occur after the initiation and the formation of the niche, which is the cancer niche where cancer survives, thrives and grows and develops. So the question that I asked the people, are these abnormal mutations that we're seeing, just because it says they're abnormal mutations in the research, doesn't actually mean that they are. They could just be necessary changes to eventually produce the cancer rogue organ, which is actually a barrier. And this is the question I need to ask people now. And new novel mRNA vaccines. An attempt to use these mRNA vaccines then to produce novel antigens of cancer cells which would warrant an immune response against cancer cells. This is where the whole immunotherapy is based on. The idea that cancer cells have novel or new antigens different from our normal cells. And the immunotherapies and the vaccine therapies are going to be against the bulk tumour cells, which are not even the main drivers of cancer. Cancer is driven mainly by cancer stem cells. And the so-called cancer novel antigens, the antigens that are expressed in these cancer cells, are found on our normal stem cells, embryonic stem cells, and even in our normal tissues and cells. And you'll find that with the reference of cancer stem cell surface markers on normal stem cells. It's different levels of expression, yes, I understand this, at different times of when cancer is initiating, progressing or metastasizing. However, looking further into these expression levels, they are transient. And what transient means is basically their expression levels of these genes changes owing to signaling pathways or morphogens. Morphogens which are basically growth factors which stimulate cell growth. For example, you have the Wnt pathway, Bohr morphogenic protein, fibroblast growth factor, and hepatocyte growth factor, and many more. I ask you all, and this is a question for everyone, what then would be the stimulus that would require the need for a rogue organ? And cancer is a rogue organ, it's not just aberrant abnormal cells proliferating out of control. What would be the need for a rogue organ to act as a barrier and a defence of cells? So I'll leave that one with you guys, and I'll speak to you all soon again.